Okay, so we will now start the fourth lecture of quantitative big imaging. And so this one will be basic segmentations and discrete binary structures. So in terms of sort of the outline, again, like most of the lectures, it will sort of start off with a little bit of the motivation, and then qualitative approaches. So what sort of approaches have been used up until now? And what approaches don't require sort of computational techniques, what can you sort of look at yourself? And then sort of the main topic for this, which is this idea of thresholding. And so, what does this mean? How do you apply threshold? How do you evaluate if it sort of works well? A little bit of more implementation and morphology. And then in the next lecture, we'll sort of cover some of this contouring, mask creation, and some of the more advanced techniques that we have. So in terms of the applications, it's quite simple. Also for this lecture, the feedback that I've gotten so far from the forum, so I do try to read it every week, is that I'm going too slow. Is that a general consensus? Should I speed up? Who thinks I should speed up? Few people. Who thinks I should slow down? A few people. I mean, so the forum is quite nice because it's anonymous, so I have no idea who says anything. And so if you send feedback there, you don't have to feel embarrassed or whatever about whatever your opinions are. But if it is too slow or most people think it's too slow, I can definitely speed it up. And if it's too fast, I can also slow it down. But yeah, I guess I'll try to stay at the same speed-ish. And for the people where it's too slow, there's, particularly in this lecture and the next lecture, there's lots of advanced exercises that are quite challenging. And so if you do those exercises, you should be kept quite busy, even if the course material isn't moving as quickly as you'd like it to. So we do try to have those options for the people who are feeling very comfortable with the material or where this is more review for them. But yeah, so just getting back to this is sort of this idea of sort of two-phase materials, which is what we'll start with. And so that's like looking, you have bones and cells, or you have, a brain and you're trying to segment where's the brain and where's the skull. So kind of where there's just two categories that you're trying to separate. And then we sort of have multiple phase materials. And so that's where you're now looking for multiple different categories. So it's not just binary. It's where it's, skull, brain, air, or, human body, organs, lungs, fluid, something like this. And so that then is sort of a step up for this. And then we'll look at sort of filling holes in materials. And then we'll get at the very end to segmenting fossils. So we'll have some examples, I think from the Planet Earth documentary by David Attenborough. Some of the samples from there were actually measured using X-ray tomography, and they segmented them. And so for the exercises today, you'll have to segment the same data sets, and you'll see how difficult it is to actually segment them really well. And we'll go over a few techniques for how you can improve that. And then kind of the last thing will be sort of attempting to segment the cortex and brain imaging if we get to that today. Again, so for sort of literature and useful references, the sort of image processing handbook that I mentioned initially, where you can get it online, they cover a lot of these topics in sort of much more detail with a much more theoretical background than we'll cover them in this class. And then in sort of models and ROC curves, which we'll cover in the second part of this lecture, there's some nice sort of talks in general about why ROC curves are important outside of the imaging space and sort of other applications to help you understand them. But we'll cover ROC curves later, so don't worry if that doesn't mean anything to you now. So in terms of motivation of kind of why do we do these experiments in the first place, and this links back to the first lecture, we were really talking about kind of what is the whole point of doing all these experiments. And so the first type of experiment we really do is exploratory. And this is really visual, qualitative. You want to look at a sample, you want to see what's inside it. You don't really have a hypothesis before you're just looking, So if you're trying to see, how many, does this animal maybe have teeth? What does this look like? How many cells are inside this sample? Is there a difference between healthy and unhealthy? So you don't really have a clear hypothesis. You don't really have knowledge or expectations. You're just trying to look at a sample. So this first type of work is really what comes in with sort of qualitative analysis. And that's where you go in as unbiased as possible. So you just try to explore and see what's present in the data. The second type you get into is this sort of to test a hypothesis. And so this is where you sort of take your exploratory information and start to come up with a hypothesis. So once you've looked at a few samples or a few dozen samples, you can then start to say it looks like the cell size between healthy and unhealthy groups decreases. And so this is where you can now make a hypothesis, and you need to start coming up with what samples do I actually want to measure, what analysis do I want to do, and then you get questions here, sort of like, does temperature affect bubble size? Is this gene important for cell shape? Does higher canal volume make bones weaker? As sort of an example for when these come in. And then sort of the last one was, does the granule shape affect battery life expectancy? So this is sort of what we're looking at. You have this sort of original image, of like a cell. This is what you might see in a textbook. And then you have sort of what we get from an imaging modality. Not sure why that doesn't show up. Guess this way works quite well. Boop. And so when we get from an imaging modality is that's a sort of like black and white image. Each pixel's sort of, some level of gray, and the sample doesn't have any labels with it. And so now it's our task to take this image and start to do something meaningful with it. So to kind of test a hypothesis, if we perform an experiment in bone to see how big the cells are inside of the tissue, we sort of have this workflow. From those of you who took the micro and nanotomography class, they should be quite familiar. That you have this bone sample, you shine x-rays through it, you do these reconstructions. You then get this sort of huge data set out, 2500 by 2500 by 2000 times 32-bit, which means you could have 56 gigabytes per sample of data that you're trying to analyze. And in sort of the first lecture, we covered, the third lecture, last week you covered this image enhancement. So you have all of these complicated techniques you can apply, these anisotropic diffusion filters, and 20 hours of compute later, you have less noisy data, but you still have a huge amount of information. So you actually haven't done anything to separate the two groups you're trying to look at. And so this is kind of where this lecture starts to fit in. So you have much nicer looking images, hopefully, but you still haven't gotten to the end result. And if we look at kind of what we wanted in the first place, it was this single number. And so maybe your task doesn't seem like it collapses to a single number, but with most analyses, what you're actually trying to get is you're trying to turn an image into a number. And so, that might be the volume fraction of canals in your image. That might be the number of cells that you have. That might be the average cell stretch or volume variability, or any number of other metrics that you could come up with. But really the goal you're trying to do with this is come up with a single number that describes your sample. And so this is where segmentation starts to help us, because right now we still have, billions of numbers, and what we want to do is collapse that down to just one. And so in sort of model-based analysis, every step we perform, simple or complicated, is related to sort of the model of the system that we were dealing with. And so this is where Occam's razor, that I guess most of you have probably heard of before, is very important, and that the simplest solution is usually the right one. And so this isn't a mathematical theory or anything. It's just, if you make a model that uses Bayesian neural networks optimized with genetic algorithms and fuzzy logic to segment your sample, that potentially isn't the right solution to your problem and introduces lots and lots of new types of error and complicated problems in your result. And so if you can use a simpler solution and it gives you almost the same results, that will be a much better way to analyze your data. And so often you'll see papers where they do really complicated things. And one of the examples that we've seen is that, they're trying to segment tumors in brains, and you look at the image, and the tumor's a bright white spot. And they train a special type of very complicated neural network to try to automatically segment these tumors. And in fact, if you looked at the image and just said you keep all the points that are brighter than 120, you would have a much better model for the tumor, and you wouldn't have a really complicated system you were trying to understand. And so what we'll do is we'll start off with the most simple techniques, and we'll show you how you can evaluate them. And then we'll sort of cover progressively more advanced techniques and see how they improve the results, with the sort of lecture 5 being the ultimate one, where the last technique we'll cover, lecture 6, where we have sort of the full neural network approach, and we can see how much that improves over sort of standard models that we have. But that the idea is that you should be able to get fairly reasonable results with simple techniques, and the more complexity you add should sort of have a clear added value in the performance of your system. And if it doesn't, then you shouldn't add that step. So if we sort of go back to filtering and enhancement, we kind of had this model before. I think Anders presented this a bit differently. But the basic idea was that we have this sort of measurement, and that our measurement is equal to our sample plus some sort of noise. So in a 2D image, each point that we measure is equal to the sample plus some sort of noise signal. And that what we want is we want this sort of image enhancement step, that when we apply this filter to the sample, we get the sample out again, so that it doesn't change anything about the sample. But when we apply it to noise, it turns it to 0. And so that when we apply it to the measured signal, what we get out is just the sample itself, and we get rid of the noise. And so the problem is that most filters, like Gaussian or median or any of these other ones, they have some effect on the real signal, but a much stronger effect on the noise. And so maybe they damage the real signal by 10%, but they decrease the noise by 90%. And so that's a good trade-off, because it gets rid of noise much more strongly than it gets rid of the original signal. But of course, what bad filters do is they get rid of much more signal than noise, and so you end up with just a noisy output. And so what you'll often see is that if you do something like sort of simple edge detection or edge enhancement on an image, you'll end up amplifying the noise a lot, and maybe not amplifying the signal that much. And so that's why often when you're using these kind of filters, you have to be very careful that they're handling the signal and the noise correctly, and they're not making noise much bigger. Because if they're making noise much bigger, then the task of sort of sorting out what signal becomes very tricky. And so now this is sort of qualitative metrics. What did people used to do? And this is looking at sort of, what comes out of the detector. So this is with this simple cell example, that we have this image and you have this histogram. And so in a very simple model, this is the absorption coefficient. White light microscopy, you have the cell, you shine white light behind it, and you basically record sort of how much is being absorbed at each point. And that's sort of the metric that you have for your image. And that you can look at it by eye and sort of do quite detailed analysis. So you can say, it has a flagellum, there's a bunch of little cilia there. It looks like there's this nucleus, which takes up, maybe a third of the volume inside or something along those lines. And that you can have some sort of basic analysis based just on sort of what you see in this image. You can also sort of start to get more quantitative. So if you look at sort of the old, the way they did gas chromatography, for example, in the 80s, was they would, print out a chart on equal, I called equal weight paper. And then you could cut out the object you were interested in. In this case, it was sort of a curve. And then you would weigh it, and that would tell you the area of the object that you were looking at. Clearly, these are things that we don't use anymore today, but those are sort of like the old techniques that people would follow in order to try to analyze samples like this. And that what we see about this is that we have the ability to sort of quantify or qualitatively describe a lot of different things in the image that we're able to see. This is a flagellum, these are cilia. This is the same color as the nuclei, but it's not connected to it, and so we count it as a separate object. And so this shouldn't be undervalued, because what's quite important is that you're able to bring some common sense and judgment to your analysis, and that when you throw it in an algorithm and it says it has 25 cilia, it's very easy to kind of blindly trust those results and say, okay, it said 25, therefore that must be right. And with the qualitative analysis and doing kind of sanity checks, you're able to look at very complicated structures and see, what makes sense in this image and what doesn't make sense in this image, and come to very reasonable conclusions. And so all of the steps we perform are sort of a balance between, looking at the images, making some assessments for yourself, and then applying a quantitative analysis to make sure that you have a robust way of quantifying the things that you already see. And so in this class, we're going to focus on primarily things that you can quantify yourself, but that are very time-consuming to do. And so we're not going to be focused on problems where, you can't see it in the image yourself. And so, for example, if you were trying, to quantify, in this image it's quite difficult, but if you wanted to figure out, how many cilia are on the backside of the cell, that's not something we can see ourselves. Maybe it's possible to make some sort of super fancy model that's able to estimate the number of cilia on the backside by changes in the curvature on the front side, but that's outside the scope of what we're doing. So what we're really focusing on are things that you're already able to do, and not trying to have a machine solve problems that you can't solve today. And so those are sort of important problems to keep distinct, because right now machines, and particularly with sort of AI, are getting much better at matching us for analyzing things that we're able to see. But they're very poor, or it's very difficult to have them do tasks where you try to have them do something a human can't do at all today. And so when it's about making humans faster or more reproducible or reliable, that seems to work fairly well. But when it's about doing things that humans can't do at all, where the signal may or may not actually even be present, then it's much more challenging. So when we go back to sort of the original approaches that we covered in the first lecture, we sort of have this naive division of image processing. And so segmentation fits very well into these different categories. And so we have kind of the experimentalist approach, the computer vision, and the deep learning. And what we'll cover is sort of in this first lecture, it will be mostly the kind of experimentalist's approach. We'll start to get into some of the computer vision. In the next lectures, we'll have the computer vision and the deep learning approaches. And so you'll see for the same kind of problem, how can you apply each different technique and what do they mean for what you're looking at? And so the model-based is, again, like probably most of your background, it's really experimentalist driven. And so you've collected the data yourself and now you need to analyze it. You have this problem-driven, top-down reality model-based. And that the machine learning approach is much more, computer vision, deep learning, and that's results-driven and technique-driven. And so the model-based analysis goes back to this sort of very simple slide from before, where you have some sort of impulse or excitation. You have a characteristic you're trying to model, the response, and the detector system. And so what the model-based analysis means is that we'll try to take things we understand about all four of these steps in order to perform the segmentation of our images. So by knowing what these processes are, how can we decide, where we mark something as being part of bone and something being part of air, for example. And so there's many different techniques, but they all sort of have this same standard mechanism that you have your measured signal, you have your system, you have your stimulus, and you have your sample. Obviously different math behind it, different coefficients, units, and mechanisms, but that's your fundamental output. And again, with sort of direct imaging, there's things like uneven illumination caused by incorrectly adjusted equipment, fluctuations in power, fluctuations in brightness, and so that you have this kind of simple model in direct imaging where the F of your system is simply A times B. And so you take your stimulus, which would be your beam profile, and your system, which would be sort of the absorption coefficient in your sample. And that what you end up with is the measured value you'll have to divide or correct for the beam profile with. And so what that looks like in sort of a very simple example with the cell before is that, if you have some sort of, laser-based system, you might end up with a beam profile that looks like this, which means when you measure something on your detector, you end up with an image like that. And so if you want to know what the original structure of the image looked like, you'll need to divide by this beam profile so that you can get this sample image back originally. And so this is a very common, probably the most common types of processing you'll do is where you're just trying to correct for the beam or the background or some other factor that's causing what's on your detector to not really match what your sample looks like. And luckily with direct imaging, this is usually as simple as a division. It just becomes more complicated if this beam is moving around or changing in shape all the time, or it's not possible to measure the beam directly. And then you'll have to come up with some more complicated approaches for doing that. So here, we're able to do the same sort of analysis where we have the beam profile shown as this line, the detector shown as the green, and the sample shown as the orange. And that we can see, in the middle, these signals match up quite well. Here the beam is fairly uniform, and so the sample and the detector overlap perfectly. But as we get closer to the edges, in sort of a single line through the image, we end up in a situation where the sample image is quite bright, so what we'd actually expect in the sample, and that what we measure at the detector is quite dark. And so in order to correct for that, we sort of need to amplify things towards the edges and keep things in the middle sort of constant. We see that there's a few parts here where the detector is actually higher than the sample. And so here we'd actually need to correct in the other direction. But that kind of shows us for one line in a sample like this, how we would go about sort of correcting for those images. And so frequently, there's fall off as the beam moves away from the center. In photography or Instagram, this is called vignetting. So where you have sort of the drop off of brightness, you get darkness towards the edges. And this is often something that you need to correct for because you want to have sort of a constant information content in your image. You don't want to characterize the lens when you're doing your analysis. So you wouldn't want to say that this cell is smaller because it's very, very dark there, because that has nothing to do with the cell, that just has to do with your imaging system. And so, and so this can make extracting detail away from the center quite challenging. And so also when you look at this, it becomes then very difficult here to see where the cell boundary actually is. That information, particularly on a projector, is then very challenging to make out. And in some cases, if the imaging is done poorly enough, is completely lost. And so when you have these problems, it's very important to know that your ability to extract information from the image is also reduced in areas where this beam profile, gets, or beam profile changes get more severe. So with absorption imaging, and so this is sort of a more specific example of this, and this is where we have a Beer-Lambert law in place, where we're looking at sort of an object that's absorbing the signal. So that's the only interaction that it's having with it. And this law basically tells us that what we see is a combination with how thick the sample is and how much the sample absorbs. And so this very commonly comes into place in X-ray imaging or cellular imaging, where you're shining a light through, and the interaction with the sample is simply absorption or attenuation. And so here we have sort of the simple model, where it's just sort of exponential decay, and so the thicker your sample is, the more it absorbs. We also end up with sort of more complicated setups where you can have changing materials. You can have materials that respond differently to different wavelengths, and so if you're using white light illumination, you might have to take that into account. But typically, we just look at sort of this exponential decay. And so you're trying to say, I can either measure the attenuation or I can measure the thickness. So if I know the attenuation, I can measure the thickness. And if I know the thickness, then I can measure the attenuation coefficient at any point in the image. And so, what we have for this is, if we look at sort of what we have on our detector, we'll end up with a curve like this. So this is the Beer-Lambert law that basically says as that attenuation coefficient goes up, the brightness on our detector goes down. So here we see the brightness on our detector, assuming a constant field, and here we change the attenuation coefficient. And then we end up with sort of, distributions of the attenuation coefficient in our sample, and then distributions of our intensity in the output. And so here, for example, these peaks are kind of equally separated, but we see in this image that the orange and green peaks kind of get smashed closer together, and that this blue peak gets spread out. And so even if the attenuation coefficients in your system are different, so you're looking at different materials, a lot of times when you're imaging them, they'll get sort of stretched or moved out, so that it's much easier to tell the difference between sort of a really light material and a medium material than it is between two dense materials, that they get much closer together. And so for your experiment design and your imaging setup, it's important to take these into account, because just the attenuation coefficient itself isn't enough to tell you how easy it's going to be to segment or threshold your images. And so now to get to a concrete example, if you look at something like mammography, and so it's an area where model-based absorption imaging is kind of problematic. And so the starting point for this is very much the constant illumination, which is rarely the case, but we'll use that for this model, and that you have this sort of exponential decay. And so what we end up with is sort of this integral of 0 to how thick the sample is, and we're able to measure the attenuation, or we're trying to get the attenuation coefficient at each position inside the image. And so if we're doing this, we have this problem that we're unable to separate the alpha attenuation, the alpha and D terms. And so if we take sort of a basic model of a breast volume as half a sphere with constant absorption factor, we can now set sort of our example alpha as 1e minus 2, and that we can turn this integral into a summation in discrete space. And so basically what this looks like is we can make this sort of phantom image. And obviously this is not a very realistic image of a breast, but this is kind of what we have. This would also be interactive in your slides as well, where we have this sort of breast mask, where it's this simple ball, radius 50, and we just take half of it. And so that what we can do for this is we can now say this is the distribution of the intensity at our detector. So that's the number of pixels that have each level of brightness. And if we were to image this from on top, this is the image that we'd expect to get. And so the density of this is constant throughout the entire sample, but we still end up with this image because this is showing us the thickness of the breast at each point. And so places where the breast is thicker, a lot more is absorbed, and places where there's no breast at all in this case are 0. And so all the light's getting through. And so if we know that alpha is constant, we can reconstruct D from the image. And so we're able to take this and directly translate it into D. And so we can say here the thickness is 100, and here the thickness is 0. And so in a very simple model case where we assume it's constant, we're able to do that, and we're able to make sort of a 3D plot showing at each point how thick we estimate the breast to be. And so this is sort of the perfect example of a very standard model for this. You can ignore the colors completely. I think that's, yeah, they're unrelated. They're just to make it easier to see. And that we are able to reconstruct that quite well. And so that's, of course, a simple problem. Obviously, breasts are much more complicated structures. And so where this starts to get more difficult is where alpha is no longer constant. And so if we change alpha to sort of, a dark lump in the center of the breast, it's impossible for us to tell if the breast is actually abnormally shaped or if there's a lump or a denser region inside it. So we're able to, add this weird little lump, which, could be a tumor, could be something else, in this very simple data set. And that we have sort of each slice 3D by 3D in this. And that we're able to apply Beer's law. And that we see, here it looks like there's this sort of constant region. And so notice we didn't change the shape of the breast at all. We just changed the attenuation at some of the points inside of it. And so now we have this sort of, our distribution changes a little bit. Our image looks quite a bit different. And that now if we try to do this reconstruction where we assume that alpha is constant, we end up with this thickness map. And that if we were to make a map of the breast, we would now see that we have this sort of block inside it. And so we know that actually the breast isn't shaped like that. It doesn't have this square inside it. In fact, there's a region inside where the density is changed. But if we are just trying to take the images and reconstruct, we don't have the ability to separate what's actually coming from the changes in attenuation and what's coming from the changes in shape. And so that's where these models become quite difficult, because those structures we now don't see. We just have this sort of compressed information where we have attenuation and thickness on top of each other. And so we can either reconstruct thickness accurately or reconstruct attenuation accurately, but we can't reconstruct both accurately at the same time. And that's one of the major challenges we deal with in 2D imaging. And so kind of what does segmentation get us? And so segmentation is able to take these images that we get and convert it to sort of a decimal value. And so this is taking these 56 gigabyte images we had before and turning them into 1.7 gigabytes. So still very large, definitely not a single value, but significantly smaller than they were before because we no longer have this sort of full spectrum of information. And the simplest way to do this is to just sort of apply a threshold in an image. And so if we start off with sort of a simple image of a cross with noise, so here we have this kind of cross image, and our goal is to segment out the cross. So the cross is the foreground and everything else is the background. We can describe the intensity with this probability density function. So those are the histograms we were showing before. And so we end up with this sort of distribution. And this shows us the number of pixels and the brightness in the image. And so that corresponds to here. We see that the cross tends to be values near 1, and that the background tends to be values near 0. And that we have, these are probably cross, and these are probably background. And so what thresholding lets us do is this is sort of the simplest segmentation method we have. And what we say is we just sort of draw a line vertically somewhere in this chart and say everything below it is part of the background and everything above it is part of the foreground. So we just make two classes out of all these values. And so here, we just see this as kind of a piecewise defined function, that if this is a greater than 0.4, it's part of the cross, and if it's less, it's 0. And so that's quite easy to apply. We see that for this image, it works perfectly. The threshold values are exactly matching the values that we want. And so there, applying just this simple criteria of it's greater than 0.4 works very well. We can also see sort of the effect of different thresholds. So if we chose 0.1, we'd end up with a ton of erroneous points. If we chose 0.3, we still have a few too many points. If we chose, a higher value like 0.9, then we'd have very few points, but we'd have sort of a guarantee that none of the points we'd have are outside the cross. And so that we always end up with these trade-offs of, if we choose a value that's too low, then we're sort of sure we're going to get every point in the cross, but we'll probably get a lot of points that aren't there. And if we have a value that's too high, we'll end up with not enough points in the cross, but fairly high confidence that we didn't get anything that wasn't supposed to be in the cross. And here, we sort of have 0.4 and 0.5, where we get the best of both worlds. So we're kind of exactly in the middle. We get everything that's in the cross and nothing that's not supposed to be into it. And this is the perfect situation, but you'll find with most samples, this very rarely actually happens. And so we'll take a more complicated example like cells. This is, again, the same sort of absorption principle we had. You have a field illuminated by white light. Cells absorb light, causing darker regions to appear in the image. Lighter regions have no cells, darker regions have cells. So we have sort of an image like this. You have all these cells in the image. We have dust in the image as well. Some of the cells are kind of clumped together, and how do we go about sort of doing a simple segmentation on this? So for anyone looking at this, how would you segment? Yeah. 180. So here. Yeah, I mean that would be a good start. Yeah. Yeah. No, I mean the left-hand side of the peak's the right one. So if we kind of look at the image, we have sort of these values here. The dark ones correspond to, 0 to maybe 100 are fairly dark. And then, 150 to 200 are bright. And so that here maybe somewhere in this range is probably the right threshold. But it's a lot more difficult to just look at this and pick the right value than it was with the other one. That, if you were to say, is 179 better than 180, it'd be quite difficult to judge that. There's not really a clear distinguishing feature that we could take on that probability distribution in order to say this is the perfect threshold, or at least I don't see an easy one to do. And so yeah, what we can do is we can take a number of different thresholds. This color does not show up very well. Can we make it? That color does not seem to work very well. So we can run, should be fairly quick to run this again so that it's a little bit easier to see. Still not great. Yeah, it's a little bit easier to see there, but it's still not fantastic. So flipping it around makes it slightly nicer. But basically we have sort of this problem that we have, if we choose a value of image is less than 100, like in this case, the right cell images less. Yeah, so if we take images less than 100 as our cutoff, then we only find sort of the really dark cells, but we don't find that much noise or other artifacts, that we're not sort of getting too many things that aren't cells inside it. And that as we increase this threshold, we sort of get up to, having a value of 200, where we definitely get every cell we wanted to get, but we also end up getting things we probably didn't want to get. So we have, a lot of this in the corner where the image is probably an illumination factor, where we then are sort of grouping all of those things together, which may or may not be meant to group together. And so we end up with this sort of challenge of which point works the best and where's sort of the perfect segmentation point. So, between 175 and 180 looks pretty good. We seem to get all the cells. We don't seem to be merging them together too much. But that becomes a very sort of qualitative assessment. And that's what we'll get to in the second part of this lecture, how we can make this a bit more robust and have a better feeling for if our technique is working well or not. We can also apply these same sort of analyses to more complicated images. So the images we've dealt with so far have just been sort of color grayscale images, and we can actually apply it to any type of image. So if we have an image where there's a vector at each point, if you're working in weather, this happens quite frequently where you get sort of wind patterns over a region of the Earth, or if you're dealing with sort of flow images and you're trying to kind of see how flow movements are happening, you can do segmentation the exact same way. And so if we have an image like this where we have at each point not just an intensity value, but also a vector indicating sort of where that intensity is going, which can be sort of visualized like this, so that you have a color associated with each point and a vector. That you can also start to apply segmentations to this the same way as you would another image, where instead of just having the intensity go into your equation, you can now look at a vector or the orientation or a combination of intensity and orientation or a huge number of different factors, so that the threshold or segmentation technique remains more or less the same, but the amount of information you can put into it goes up. And so here we see that we can segment all the arrows that are pointing to the upper right direction by keeping all the ones where the x component of f of x and y is greater than 0.25. And so that just gives us the components that are moving up and to the right. And so this allows us to take our sort of complex stream image and turn it into a simple grayscale black and white image that we can do something else with. We can also look at this as sort of a joint probability distribution, where we can then pick sort of the region of this. So this image doesn't have a simple 1D histogram. Instead, it has sort of joint distributions because you have a number of different factors at each point. And so there's a lot more things you need to take into account. You can also apply sort of more complicated thresholds where you focus on the values that are pointing upwards, or you look at orientation, or any number of other sort of approaches with that. So are there any questions on that original threshold material? Okay. So we'll take, I guess, a short break, and then we'll move on to the second part of the lecture, which is in different slides because the 3D images made this too big to have in one slide deck.